Beneath the surface, something is deciding. Right now, in oceans across the planet, animals without brains are making choices. They're tracking time, responding to threats, coordinating with neighbours they've never consciously met. They remember the rhythm of tides, anticipate the return of light, navigate chemical gradients invisible to us. They've been doing this for 600 million years. We thought brains were required for thought. We were wrong. But how can something without neurons make decisions? How can an animal with no central processor coordinate thousands of cells into unified action? How can a creature that never moves through space still navigate time? What if intelligence isn't what we thought? Not consciousness or cognition or internal monologue, but something more fundamental, written in the oldest language of life itself: signal, response, memory. Tonight, we're diving into the silent minds of corals and their relatives. We'll trace electrical impulses through jellyfish bells, watch sea anemones wage chemical warfare, and discover what happens when memory exists without a brain to hold it. By the end, you might never think about thinking the same way again. Because if corals can decide without brains, then perhaps consciousness isn't a threshold we crossed. Perhaps it's a gradient, an ancient dialogue between cells that's been running since before eyes opened, before blood flowed, before the first synapse fired in the first brain. So let's begin where all intelligence begins, with the simplest question a living thing can ask. Is this safe, or should I close? This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a coral polyp in shallow water. It's the size of a pinhead, translucent, almost invisible against the calcium skeleton it's built around itself. Tentacles wave slowly in the current, armed with stinging cells that fire on contact. It has no eyes, no brain, no central nervous system, just a tube with a mouth at one end and tissue stretched between. And yet, it's making decisions. As dawn breaks and light filters through the water, something shifts inside the polyp. Not thought, not intention, but change. Proteins in its cells respond to photons, triggering cascades of chemical signals. The tentacles retract slightly. Inside the polyp's tissue, microscopic algae called zooxanthellae begin photosynthesizing, converting light into sugar. The coral doesn't control this directly, it can't, but it responds to it. Throughout the day, as light intensifies, the polyp adjusts. At night, when the algae stop producing energy, the tentacles extend fully, reaching into darkness to capture drifting plankton. The polyp has no clock, no calendar, no concept of time as we understand it. But it knows day from night. It anticipates dawn. It prepares for dusk. Somehow, without a brain, this tiny animal is tracking the rotation of the Earth. This is the paradox we're about to explore. For most of human history, we believed intelligence required complexity. We drew a line between thinking things and reactive things, between minds and mechanisms. At the top, we placed ourselves, conscious, aware, deliberate. Below us, other mammals, then birds, reptiles, fish, insects. And at the bottom, the simplest animals, sponges filtering water, jellyfish drifting with currents, corals anchored to rock. We called them primitive. We thought they were barely alive, just reflexes wrapped in tissue, automatons running genetic programs with no awareness, no choice, no intelligence. But that line is dissolving. Over the past 2 decades, research has revealed something stunning about these ancient brainless creatures. They're not simple. They're not mindless. They're making decisions that would challenge organisms with far more complex nervous systems. They're responding to environmental cues with precision that suggests not just reaction, but integration, the processing of multiple signals simultaneously to produce adaptive behavior. According to work by neurobiologist Raphael Yuste at Columbia University, and his colleagues studying neural systems across the animal kingdom, the fundamental mechanisms of decision-making, signal integration, threshold responses, memory storage, don't require brains at all. They emerge from the basic properties of cells communicating with each other. Neurons evolved from this ancient cellular communication system, not the other way around. The brain is an optimization, a refinement. But the intelligence it enables? That's older than brains by hundreds of millions of years. Corals are part of Cnidaria, a phylum that includes jellyfish, sea anemones, and hydroids. They diverged from the rest of the animal kingdom roughly 600 million years ago, before the Cambrian explosion, before predators evolved, before eyes or ears or centralized nervous systems existed. They represent one of evolution's earliest experiments in multicellular animal life, and they've been phenomenally successful. Coral reefs cover less than 1% of the ocean floor, but support 25% of all marine species. They've survived 5 mass extinctions. They've adapted to every ocean, every depth, every temperature range that can support animal life. Not bad for something without a brain. But here's what makes them particularly fascinating. They're the simplest animals with a nervous system. Not a brain, not a central processing unit, but a nerve net, a distributed network of neurons spread throughout their body with no clear center, no hierarchy, no command structure. It's democracy at the cellular level. And somehow, it works. When a coral polyp retracts its tentacles, thousands of cells are coordinating. Muscle cells contract. Sensory cells detect touch or chemical signals. Neurons transmit electrical impulses across the body. Cnidocytes, the specialized stinging cells unique to this group, fire or remain dormant based on molecular cues. All of this happens in milliseconds, without a brain directing traffic. The question is, how? How does an animal with no centralized control integrate information from multiple sources and produce coherent, adaptive responses? How does it distinguish between a harmless piece of debris and a predator? How does it coordinate feeding with neighbors to avoid competition? How does it time its reproduction to mass spawning events that happen once a year, triggered by moon phases and water temperature? These are the kinds of problems we assume require significant computational power, yet corals solve them with a nervous system so simple it barely qualifies as a nervous system at all. And they're not alone. Sea anemones, close relatives of corals, exhibit behaviors that border on the bizarre. They engage in territorial combat, launching stinging cells at competitors. They form symbiotic relationships with specific species of fish and shrimp, recognizing partners and defending them from predators. Some species can move, slowly, across the seafloor, relocating when conditions deteriorate. They remember environmental conditions and adjust their behavior accordingly. Jellyfish, perhaps the most alien of all sea cnidarians, navigate ocean currents with startling precision. Box jellyfish hunt actively, tracking prey with image-forming eyes despite having no brain to process visual information. Their nerve net somehow extracts enough information from their 24 eyes to guide hunting behavior that's more sophisticated than anything a reflex could produce. These animals are performing feats of biological computation that we're only beginning to understand, and, in understanding them, we're starting to realize something profound. Intelligence isn't binary. It's not something you either have or don't have. It's a spectrum, and it goes back much further than we thought. Tonight, we're going to explore that spectrum from the bottom up. We'll start with the nerve net itself, what it is, how it works, why it's so different from our own nervous systems. Then we'll look at specific behaviors, decision-making, memory, coordination, even learning. We'll examine the molecular mechanisms that enable these behaviors, the evolutionary history that shaped them, and the philosophical implications of what it all means. Because if corals can think without brains, if intelligence predates the structures we assumed were necessary for it, then we need to rethink not just what intelligence is, but where it came from, how it evolved, and what other forms it might take, here on Earth and perhaps beyond. So let's dive deeper. Let's follow the signals moving through tissue that's been making decisions since before brains existed. Let's listen to what the oldest minds on Earth have been saying all along. Let's begin at the beginning, with a nerve net older than memory itself. Maybe intelligence was never about having a center. Let's start with what a nerve net actually is, because it's not intuitive. When you think of a nervous system, you probably picture something like your own. A brain at the top, a spinal cord running down your back, nerves branching out to every part of your body. Information flows inward to the brain processes it, decisions get made, commands flow back out. It's hierarchical, centralized. There's a clear chain of command. A nerve net is nothing like that. Imagine instead a fishnet laid flat across a table. Every knot in that net is a neuron. Every thread connecting the knots is a synapse. There's no center, no hub, no command post. Every neuron connects to the neurons around it, and they connect to their neighbors, and so on, spreading out across the entire body of the animal in a loose, distributed web. Now imagine that net is alive. That signals can start anywhere in the network and propagate outward in all directions simultaneously. That each neuron can both send and receive, with no distinction between sensory neurons, processing neurons, and motor neurons. Everything is everything. That's a nerve net. It's one of evolution's earliest solutions to a fundamental problem. How do you coordinate a multicellular body? How do you get thousands or millions of cells to work together, to respond to threats, to capture food, to reproduce? Single-celled organisms don't need coordination. They are the decision maker and the actor simultaneously. But the moment you become multicellular, you need communication. You need some way for a signal detected in one part of the body to influence what happens in another part. The nerve net is that solution, stripped to its absolute minimum. According to research published in 2020 by evolutionary neurobiologist Detlev Arendt and his team at the European Molecular Biology Laboratory, nerve nets likely evolved before brains, before centralized nervous systems, possibly as early as 700 million years ago, in the last common ancestor of all animals with nervous systems. The structure is beautifully simple. Neurons connected to other neurons, forming a continuous network throughout the body. But simplicity doesn't mean stupidity. A coral polyp's nerve net contains thousands of neurons, each one capable of firing electrical signals called action potentials, the same basic electrical impulses your brain uses. When a stimulus touches the coral surface, sensory cells detect it and trigger neurons nearby. Those neurons fire, sending electrical signals to their neighbours, which fire in turn, creating a wave of activation that spreads through the tissue. It's like dropping a stone in a pond. The ripples move outward from the point of impact, propagating through the network until they either fade or reach the edge of the polyp's body. But here's where it gets interesting. Not all signals propagate the same way. Some signals are weak. They might activate a few nearby neurons and then die out, never reaching the whole body. These are local responses. A tentacle twitches when something brushes against it, but the rest of the polyp doesn't react. Other signals are strong. They trigger a cascade that spreads throughout the entire nerve net, causing the whole animal to contract, the mouth to close, all tentacles to retract simultaneously. The difference between these responses isn't random. It's determined by the intensity of the stimulus, the type of sensory cell that detected it, and the state of the nerve net itself. The network is performing a calculation. Is this stimulus strong enough, dangerous enough, important enough to warrant a full-body response? That's a decision, not a conscious one. There's no deliberation, no weighing of options, no internal debate. But it is a discrimination between different inputs and a selection of different outputs based on that discrimination. The nerve net is integrating information, combining signals from multiple sensory cells across the body surface, and producing behavior that's appropriate to the situation. Research by neurophysiologist George Mackey, who spent decades studying cnidarian nervous systems, demonstrated that these networks can perform surprisingly sophisticated computations. In experiments with sea anemones, Mackey showed that the timing of signals matters. 2 weak stimuli delivered close together in time will produce a stronger response than either stimulus alone. The nerve net is summing inputs, adding them up over short time windows. It's also filtering. A continuous, unchanging stimulus, like water flow, produces a brief initial response that quickly fades. The nerve net habituates, stops responding to stimuli that don't change. But a sudden change in that stimulus, a shadow passing overhead, a chemical plume in the water, triggers immediate activation. The network is distinguishing between signal and noise. It's prioritizing novelty over constancy. It's extracting the information that matters from a flood of sensory input. And it's doing all of this with no central processor, no brain region dedicated to sensory integration, no executive function making decisions. The computation is distributed. It emerges from the collective behavior of thousands of neurons, each following simple rules, each responding to its immediate neighbors. This is what computer scientists call emergent complexity, sophisticated behavior arising from simple components following simple rules. The individual neurons aren't smart. They can't solve problems alone, but connected in a network, allowed to influence each other, they produce something that looks remarkably like intelligence. The implications are profound. For most of neuroscience's history, we've studied brains by trying to understand what different regions do. The visual cortex processes sight. The motor cortex controls movement. The hippocampus stores memories. We've mapped these functions, traced the connections, built models of how information flows through these specialized structures. But nerve nets don't work that way. There are no specialized regions. Every part of the network can potentially do everything. The computation isn't localized. It's everywhere and nowhere simultaneously. And yet it works. It's worked for 600 million years. It's supported some of the most successful animal groups on the planet. What nerve nets are showing us is that you don't need complexity to produce adaptive behavior. You don't need a brain to make decisions. You need communication. You need integration. You need a way for signals to flow through a system and produce coordinated responses. The brain, with all its elaborate structure and specialized regions, is an optimization. It's faster, more flexible, capable of far more sophisticated computations than any nerve net. But it's not a prerequisite for intelligence. It's one solution among many to the problem of coordination. And in some ways, nerve nets have advantages that brains don't. They're robust. Damage a brain, and you lose function, sometimes catastrophically. Damage a nerve net, and the rest of the network continues functioning. Cut a coral polyp in half, and both halves will regenerate, each building its own complete nerve net. There's no critical structure to lose, no single point of failure. They're scalable. A coral colony can grow to enormous size. Some are hundreds of years old, spanning meters, and the nerve net grows with it, maintaining function across the entire structure. There's no equivalent in centralized nervous systems. Your brain can't just keep growing indefinitely. They're democratic. In a hierarchical nervous system, higher centers can inhibit or override lower centers. But in a nerve net, every neuron has an equal vote. The behavior that emerges is genuinely collective, the sum of all signals across the entire network. Of course, nerve nets have limitations too. They're slow compared to brains. Signal propagation speeds in cnidarian nerve nets are roughly 1 to 10 meters per second, compared to up to 120 meters per second in human nerves. They can't support complex behaviors like planning or abstract reasoning. They can't store long-term memories the way a hippocampus can. But for the problems corals need to solve, when to feed, when to retreat, how to respond to predators and competitors and environmental stresses, nerve nets are sufficient. More than sufficient. They're elegant. Still, the question remains, if the hardware is so simple, if there's no central processor making decisions, how do corals manage to produce behaviors that look so much like decision-making? The answer lies not in the neurons themselves, but in what they're connected to. The first conversations were chemical. Long before the first neuron ever fired an action potential, cells were talking to each other. They had to be. The moment life became multicellular. Roughly 800 million years ago, maybe earlier, individual cells lost their autonomy. They became part of something larger, and that meant they needed to coordinate. The question is, how do cells without nervous systems communicate? The answer is everywhere, hiding in plain sight. It's in your body right now, in every tissue, every organ. Cells release chemicals into the space between them, and neighboring cells detect those chemicals with receptor proteins embedded in their membranes. When a receptor binds its target molecule, it changes shape, triggering cascades of chemical reactions inside the cell. Genes switch on or off, proteins get manufactured or destroyed, the cell's behavior changes. This is signal transduction, and it's older than animals. Plants use it, fungi use it, even bacteria use versions of it to coordinate behavior in biofilms. It's the fundamental language of cellular life. Corals use it constantly. Inside a coral polyp, cells are releasing chemical signals called neuropeptides, nitric oxide, and calcium ions. These molecules diffuse through tissue, binding to receptors on neighbouring cells. Some signals are short-range, affecting only cells a few micrometers away. Others spread further, creating gradients across the entire polyp. According to research by molecular biologist Eldon Ball at Australian National University, who spent years studying gene expression in corals, these chemical signalling pathways are remarkably similar to the ones used by organisms with much more complex nervous systems. The same molecular machinery that regulates neuron communication in your brain exists in corals, just deployed differently. For example, in your brain, when one neuron wants to signal another, it releases neurotransmitters across a synapse. The receiving neuron detects those neurotransmitters with receptors, and if enough receptors are activated, the neuron fires its own action potential. The signal passes from cell to cell in a chain. In coral nerve nets, the process is nearly identical. Neurons release neurotransmitters. Neighbouring neurons detect them. Electrical signals propagate through the network. But here's what's fascinating. The same neurotransmitters that neurons use to talk to each other are also used by non-neuronal cells. Muscle cells use them. Epithelial cells lining the coral surface use them. Even the symbiotic algae living inside coral tissue respond to some of these signals. The nerve net isn't a separate system. It's embedded in a body-wide chemical communication network that predates neurons entirely. Let's make this concrete with an example. When a coral polyp detects a predator, say a fish nibbling at its tentacles, sensory cells in the epidermis release glutamate. This is the same neurotransmitter your brain uses for excitatory signaling, the chemical that makes neurons more likely to fire. In the coral, glutamate does several things simultaneously. First, it activates nearby neurons in the nerve net, triggering the electrical cascade that spreads through the body, but it also binds to receptors on muscle cells, causing them to contract even before the electrical signal reaches them. And it triggers cnidocytes, the stinging cells unique to cnidarians, to fire their harpoon-like threads into the predator's tissue. One signal, multiple effects, coordinated across different cell types. This is crucial to understand. The intelligence of corals isn't localized in their nerve net. It's distributed across their entire cellular architecture. The nerve net coordinates and amplifies, but the decision-making, the discrimination between different inputs and the selection of appropriate responses happens at multiple levels simultaneously. Research published in 2019 by neuroscientist Pavel Burkhardt at the University of Bergen revealed something even more remarkable. He and his team studied the closest living relatives of animals, single-celled organisms called chionoflagellates. These microscopic creatures look like miniature versions of the collar cells that line sponge bodies, a single cell with a whip-like flagellum surrounded by a collar of finger-like projections. Chionoflagellates have no nervous system. They're single cells. But Burkhardt's team found that they possess many of the same signaling proteins that neurons use to communicate. They have receptors similar to neurotransmitter receptors. They have proteins involved in forming synapses. They even have ion channels that can generate electrical signals. In other words, the molecular toolkit for building nervous systems existed before nervous systems did, before animals, before multicellularity. These proteins weren't originally for neural communication. They were for basic cellular functions, sensing the environment, regulating internal chemistry, coordinating the cell's response to food or threats. When cells started working together in multicellular bodies, evolution didn't invent new communication systems from scratch. It repurposed the systems that already existed. Neurons are specialized cells that evolved to be really good at signaling. But the signaling itself? That's ancient. That's universal to cellular life. This explains something puzzling about coral behavior, how they can produce coordinated responses even when their nerve net is experimentally blocked. In studies where researchers applied chemicals that prevented neurons from firing, corals still responded to some stimuli. More slowly, less dramatically, but they still responded, because the nerve net is just one layer. Underneath it, the cells themselves are still communicating through direct chemical signaling, through gap junctions that allow molecules to pass directly from one cell to another, through mechanical forces transmitted through the extracellular matrix. The body has redundancy, multiple ways to coordinate, multiple channels of communication running in parallel. Let's zoom in further to the molecular level to see how these signals actually work. When a coral cell detects a threat, maybe a toxin released by a competing coral nearby, receptor proteins on its surface bind the toxin molecules. This binding causes the receptor to change shape, which activates a protein inside the cell called a G protein. The G protein in turn activates an enzyme that produces a molecule called cyclic AMP, or CAMP for short. Now here's where it gets interesting. CAMP is a second messenger, a molecule that carries signals inside cells. When its concentration rises, it activates other proteins called kinases, which then activate other proteins, which activate others, in a cascade that amplifies the original signal exponentially. One receptor binding one toxin molecule can trigger the production of thousands of CAMP molecules, which activate thousands of kinases, which affect millions of target proteins. It's signal amplification, turning a whisper into a shout. But CAMP doesn't just stay in one cell. It can diffuse through gap junctions into neighbouring cells, spreading the alarm. Within seconds, cells across the entire polyp are responding to a threat that only one cell initially detected. This is distributed sensing. No single cell needs to have the complete picture. Each cell responds to local conditions and passes information to its neighbours. The collective response emerges from these local interactions. According to work by cell biologist Nicole Dubilier at the Max Planck Institute for Marine Microbiology, this kind of chemical coordination is remarkably sophisticated. Corals can distinguish between different types of threats based on the specific combination of signaling molecules released. A predator attack triggers a different chemical profile than physical damage from a storm. Bleaching stress from high temperatures produces yet another signature. The polyp isn't just reacting, it's categorizing threats, matching responses to the specific nature of each challenge. And here's the most remarkable part. These chemical signals persist. They don't just flash and disappear like electrical signals in neurons. Some signaling molecules remain in tissue for minutes or even hours, creating a kind of short-term memory at the cellular level. If a coral is attacked repeatedly in the same location, the cells in that area become sensitized. The next attack triggers a faster, stronger response. The tissue has learned, in a sense, that this particular spot is vulnerable. This is habituation and sensitization, the same basic forms of learning that neuroscientists study in animals with complex brains, occurring in an organism with only a nerve net. But we still haven't answered the core question. How does this translate into decision-making? How does chemical chatter between cells become choice? The answer lies in thresholds and integration. Every cell has internal thresholds, concentrations of signaling molecules that must be exceeded before the cell changes its behaviour. When multiple signals arrive simultaneously from different sources, they add up. If the sum exceeds the threshold, the cell acts. If not, it remains quiescent. This is analog computation. The cell is integrating multiple inputs, weighing them against each other, and producing an output that depends on the total signal strength. Now scale that up. Thousands of cells, each performing its own integration, each influencing its neighbours. Signals flowing through tissue, reinforcing in some places, cancelling in others. Patterns emerging from the interaction of simple local rules. The coral polyp becomes a computational substrate. Information flows through it the way water flows through soil, following paths of the least resistance, pooling in low spots, finding its way through the network. And somewhere in that flow, decisions crystallise, not as thoughts, not as conscious choices, but as patterns of cellular activity that produce coherent adaptive behaviour. The polyp closes, or it doesn't. It releases gametes, or it waits. It extends its tentacles into the current, or it pulls them back. Each choice emerges from millions of molecular interactions, none of which alone constitutes a decision, but together, collectively, they are deciding. This is what intelligence looks like at its most fundamental level. Not consciousness, not awareness, just information processing that produces behaviour adapted to circumstance. But there's more. Because corals don't just respond to the immediate present. They track time, they remember, and that requires something beyond chemical signalling. Time is the dimension corals navigate best. You can't see it happening. A coral polyp looks essentially the same at noon as it does at midnight, but inside, at the molecular level, the clock is running. Genes switch on and off in waves, proteins accumulate and degrade. The entire cellular machinery oscillates with a rhythm that matches the rotation of the Earth. This is the circadian clock, and it's one of the most ancient and universal features of life. Cyanobacteria have it, plants have it, fungi have it. Every animal studied so far, from fruit flies to humans, operates on roughly 24-hour cycles governed by the same core genetic machinery. Corals are no exception. Research published in 2021 by chronobiologist Oren Levy at Bar Ilan University in Israel revealed that coral polyps maintain robust circadian rhythms, even in constant darkness, even when removed from the ocean and placed in laboratory tanks with no environmental cues. The clock keeps ticking, counting hours, anticipating dawn. But why? What advantage does a clock provide to an animal that can't move, that lives in the same spot its entire life? The answer is prediction. A circadian clock allows an organism to prepare for predictable changes before they happen. Corals don't wait for dawn to start photosynthesizing. Their symbiotic algae gear up hours beforehand, building the molecular machinery needed to capture light efficiently the moment the sun rises. They don't wait for dusk to extend their feeding tentacles. They begin the process in late afternoon, positioning themselves to capture zooplankton that become active at night. This is anticipatory behavior. The coral is using information from the past, the regular cycling of day and night, to make predictions about the future. It's living slightly ahead of the present moment, always preparing for what comes next. According to Levy's research, approximately 10 to 20% of all genes in coral genomes show circadian patterns of expression. That's thousands of genes, switching on and off in coordinated waves throughout the day and night. Some genes peak in the morning, others at noon, others at midnight. Each temporal window corresponds to different physiological processes. Morning, genes involved in photosynthesis ramp up. The algae inside the coral tissue need to be ready when light arrives. Midday, genes for antioxidant production peak. Photosynthesis generates reactive oxygen species that can damage cells, so defenses need to be at maximum strength when light intensity is highest. Evening, genes for calcification, the process of building the calcium carbonate skeleton, become active. This happens at night because calcification interferes with photosynthesis, so the two processes are temporally separated. Night, genes for digestion and feeding peak. The coral extends its tentacles ready to capture prey. Each process is scheduled, timed to occur when conditions are optimal. The clock coordinates metabolism across the entire organism, ensuring that different cellular functions don't interfere with each other, that resources are allocated efficiently throughout the day. But here's what makes this remarkable. The clock isn't just tracking time, it's tracking seasonal time. Corals reproduce once a year, sometimes only once every few years. The timing is extraordinarily precise. In many species, mass spawning events occur on a single night, within a few hours of each other, synchronized across entire reefs. Millions of polyps release eggs and sperm simultaneously, turning the water into a blizzard of gametes. How do they know when? The triggers are environmental, water temperature, day length, moon phase, but the coral has to integrate these cues over weeks or months. It has to measure the gradual warming of water as summer approaches. It has to count the lengthening of days. It has to track lunar cycles, noting when the full moon occurs. Research by marine biologist Andrew Hayward at the Australian Institute of Marine Science demonstrated that corals use multiple cues simultaneously. Temperature provides the rough window. Spawning happens during the warmest months. Day length refines the timing. It occurs after the summer solstice in most species, and lunar phase triggers the exact night, typically a few days after the full moon. But the coral doesn't have a brain to remember 3 days after full moon. It doesn't have a calendar marking August 15th. What it has is a molecular counting system. According to recent work by molecular chronobiologist Mikhail Matz at the University of Texas, corals possess gene regulatory networks that can track multi-day cycles. Certain proteins accumulate slowly over lunar months, reaching threshold concentrations that prime the organism for spawning. When the right combination of temperature, day length, and protein concentration occurs, the spawning program activates. The coral has been counting, integrating signals over time, building toward a decision that will execute on a single night. This is temporal computation. The organism is performing a calculation that extends across months, integrating multiple environmental variables, producing an output, spawn now, that requires exquisite timing to succeed. Because spawning synchrony matters. Coral eggs and sperm are released into the water, where fertilization happens externally. If your neighbours spawn a week before you, your gametes will find no partners. If you spawn a week late, same problem. The only way this strategy works is if everyone spawns together simultaneously, overwhelming predators and maximising the chance that egg meets sperm in the vastness of the ocean. Evolution has selected for organisms that can tell time with extraordinary precision. Not conscious time, not subjective time, but biological time measured in molecular oscillations and threshold concentrations. And the implications extend beyond reproduction. Corals also track tidal cycles, preparing for the twice-daily changes in water flow and sediment load that tides bring. Some species show clear behavioural patterns linked to tidal rhythms, extending tentacles more during high tide when plankton concentrations are higher, retracting during low tide when sediment is more likely to settle on them. They track seasonal patterns in storm frequency, growing thicker skeletons during months when wave action is most intense. They track long-term climate patterns, adjusting growth rates in response to El Niño cycles that alter water temperatures and nutrient availability. These aren't conscious adaptations. The coral doesn't think a storm is coming. But over evolutionary time, individuals whose molecular clocks happen to trigger storm-resistant growth patterns at the right seasonal windows survived better, reproduced more, passed those timing mechanisms to their offspring. The genome has encoded statistical patterns from the environment. The clock reads those patterns out, matching internal rhythms to external cycles. This is sophisticated temporal intelligence operating without any awareness whatsoever. But even more striking than the coral's ability to track time is its ability to adjust its clock when the environment changes. In experiments conducted by marine biologist Lean Bay at the Australian Institute of Marine Science, corals were subjected to shifted light-dark cycles in the laboratory, essentially giving them jet lag. Within 3 to 5 days, the molecular clock was synchronized to the new schedule. Gene expression patterns shifted, peak activity times moved. The entire temporal program rewrote itself to match the new reality. The clock is flexible, it learns. More remarkably, different parts of the same coral colony can run on slightly different schedules. In branching corals, where some branches are shaded and others exposed to full sun, the shaded branches' clocks drift earlier, while exposed branches' clocks drift later. Each part of the colony is fine-tuning its rhythm to local light conditions. This shouldn't be possible. These branches are connected. They share the same genome, the same nerve net, the same chemical signaling networks, and yet they're maintaining distinct temporal programs simultaneously. According to research by systems biologist Daniel Witchick at Boston University, this happens because circadian clocks are inherently cell autonomous. Each cell has its own clock, its own oscillating gene expression. In animals with centralized nervous systems, the brain coordinates these cellular clocks, imposing a unified rhythm on the body. But in corals, there's no central coordinator. Each cell, each group of cells, each branch of the colony runs its own clock, coupled loosely to neighbours but fundamentally independent. It's distributed timekeeping, democracy extended to the temporal dimension. And this leads to something unexpected. Corals can have memories that last longer than any single molecular signal. Consider what happens when a coral experiences a heat stress event. Water temperatures rise, the symbiotic algae produce excess oxygen radicals, the coral bleaches, expelling its algae to avoid being poisoned. This is an immediate crisis response, but after the water cools and the coral recovers, something persists. According to work by molecular ecologist Holly Putnam at the University of Rhode Island, corals that have experienced bleaching once show altered gene expression patterns for months afterward, sometimes years. Stress response genes remain slightly more active. Heat shock proteins are produced at higher baseline levels. The organism is primed, ready to respond faster if heat stress returns. This is a form of memory, not stored in neurons, not conscious or recallable, but real nonetheless. The coral has changed because of experience. It remembers in the only way a brainless animal can remember, by altering its molecular state in ways that persist across time. The mechanism involves epigenetic modifications, chemical tags added to DNA that change how genes are expressed without changing the DNA sequence itself. These modifications can last for months, sometimes persisting through cell division, effectively encoding experience into the genome's regulatory landscape. The coral's past influences its present. Experience shapes response. The organism is not just reacting to the immediate moment, it's integrating history into its decision-making. Time, it turns out, is just another dimension of information, and corals process temporal information with the same distributed molecular machinery they use for everything else. But time isn't the only variable corals track. They also navigate chemical space, distinguishing between thousands of different molecules in their environment. And that requires a sensory sophistication we're only beginning to understand. The ocean is a chemical library, and corals can read it. Every organism leaves a signature in the water. Predators release metabolites that betray their presence. Prey animals shed cells and mucus. Competing corals secrete toxins to defend their territory. Bacteria bloom and die, releasing their contents into the current. Even sunlight creates chemical changes, breaking down organic molecules, generating reactive compounds. A coral polyp fixed to its spot on the reef can't escape threats or pursue opportunities. It can't move toward food or away from danger, but it can sense what's coming. It can taste the water flowing past and respond to what it detects. This is chemosensation, and it's the dominant sense for most marine life. Vision matters in clear water, but chemicals work everywhere. In darkness, in murky water, around corners, across distances. A single molecule released by a predator can diffuse through the ocean for meters or even kilometers, carrying information about presence, identity, and proximity. Corals detect these molecules with receptor proteins embedded in the membranes of their sensory cells. According to genomic studies by marine molecular biologist Monica Medina at Pennsylvania State University, coral genomes encode hundreds of different chemoreceptors, each tuned to bind specific molecular shapes. Some detect amino acids, the breakdown products of proteins that indicate nearby food. Others detect glycoproteins specific to predatory fish mucus. Still others recognize the defensive chemicals released by competing corals. Each receptor is a question asked of the environment. Is food near? Is danger approaching? Are competitors encroaching? The answers arrive as molecules binding to proteins, triggering the cascades of cellular communication we've already explored. But sensing isn't enough. The coral needs to discriminate. It needs to distinguish between different chemicals, different concentrations, different combinations. It needs to extract meaning from molecular noise. Let's look at how this works with a specific example. Coral larvae. Adult corals are sessile, stuck in place, but their larvae swim. For hours or days after spawning, coral larvae drift with currents, searching for a place to settle. This is the most critical decision in a coral's life. Where to attach and begin building a colony. Choose wrong. Too deep, too shallow. Too exposed to sand, too close to aggressive. Competitors, and you die. The larva has no eyes, no brain, just a tiny ball of cells with cilia for swimming and a nerve net for coordination. And yet it makes this decision with remarkable precision. Research by marine ecologist Peter Edmonds at California State University Northridge has shown that coral larvae actively search for specific chemical cues. They're attracted to certain types of crustose coralline algae, pink, rock-like algae that grow in patches on the reef. These algae release compounds that the larvae detect and swim toward. Why these algae? Because they grow in locations with the right light levels, the right water flow, the right stability. The algae are indicators, proxies for habitat quality. Evolution has wired the larvae to trust this signal. But here's what's remarkable. The larvae don't just respond to a single chemical. They integrate multiple cues simultaneously. They're attracted to coralline algae, yes, but they're also repelled by chemicals released by harmful bacteria, by toxins from allelopathic seaweeds, by compounds indicating high sedimentation. They're performing a multidimensional chemical analysis, weighing positive signals against negative ones, computing a kind of habitat quality score. According to work by coral reef ecologist Mark Hay at Georgia Institute of Technology, the larvae can detect concentration gradients and swim up or down those gradients toward higher or lower concentrations. This is chemotaxis, directed movement in response to chemical signals. It requires the larva to compare concentrations on opposite sides of its tiny body, detecting differences of just a few molecules per cubic millimetre of water. The larva is navigating chemical space with precision that rivals any organism with a sophisticated brain. And once it settles, once it metamorphoses into a polyp and begins building its skeleton, the chemical sensing doesn't stop. It intensifies. Adult corals use chemosensation to detect predators. Crown-of-thorns starfish, which feed on coral tissue, release specific compounds as they digest their meals. Nearby corals detect these compounds and respond by increasing mucus production, by ramping up production of defensive chemicals, sometimes even by accelerating growth to outpace tissue loss. According to research by marine chemical ecologist Carly Kenkel at the University of Southern California, corals can distinguish between the chemicals released by different predator species and tailor their responses accordingly. A fish that nibbles tentacles triggers one defensive program. A starfish that dissolves tissue triggers a different program. A boring sponge that tunnels through the skeleton triggers yet another response. Each threat has a chemical signature. Each signature activates a specific defensive strategy. The coral is performing pattern recognition at the molecular level. But the most sophisticated chemical sensing involves interactions between corals themselves. Reefs are battlegrounds, space is limited, and every coral is competing with every other coral for access to light and plankton. When two coral colonies grow close enough to touch, chemical warfare begins. Some species produce sweeper tentacles, specialized elongated tentacles packed with stinging cells. These tentacles extend at night, reaching toward neighboring colonies, delivering venoms that kill tissue on contact. Other species secrete toxins directly into the water, creating chemical halos around themselves that inhibit competitors' growth. But before the fighting starts, there's sensing. Each coral needs to know, what species is my neighbour? How aggressive is it? How much of a threat does it pose? According to studies by coral ecologist Nanette Chadwick at Auburn University, corals can identify the species of their neighbours through chemical recognition. They detect species-specific compounds in the mucus and tissue of adjacent colonies. Based on this chemical identification, they adjust their behaviour. If the neighbour is the same species, the response is muted. There's less investment in defensive structures, less production of toxins. The colonies may even fuse together if they're genetically similar enough, sharing resources and coordinating growth. If the neighbour is a different species, the response depends on relative competitive ability. If it's a species known to be aggressive, the coral invests heavily in defence, growing faster to claim space, producing more sweeper tentacles, secreting higher concentrations of allelopathic compounds. If it's a species that's typically less competitive, the coral maintains normal growth and defense levels. This is strategic decision-making. The coral is assessing its competitive environment and allocating resources accordingly. It's distinguishing self from other, kin from stranger, threat from non-threat, all through chemical sensing. The molecular mechanisms underlying this recognition are still being unravelled, but research by immunologist Virginia Weiss at Oregon State University suggests that corals use proteins similar to the immune recognition proteins in vertebrate animals. These proteins bind to molecular patterns on the surface of other organisms, triggering signalling cascades that lead to acceptance or rejection. The coral's body is performing immunological surveillance, constantly asking, is this self or other, friend or foe, safe or dangerous? But chemical sensing extends even deeper into the relationship between corals and their symbiotic algae. Inside every coral polyp's cells live thousands of dinoflagellate algae called symbiodinium. These algae photosynthesize, producing sugars that they share with their host. In return, the coral provides the algae with nutrients and a safe place to live. The relationship is ancient. It evolved over 200 million years ago, and it's essential. Without their algae, most corals starve, but the relationship is also fragile. When temperatures rise too high, the algae produce toxic oxygen radicals. The coral, sensing danger, expels them. This is bleaching, and it's a life or death decision. Keep the algae and risk being poisoned. Expel them and risk starvation. According to research by symbiosis biologist Monica Medina, corals sense their algae's health through chemical signals. Healthy algae release compounds that suppress the host's immune response, maintaining the partnership. Stressed algae fail to produce these signals, or worse, they release distress compounds, damage proteins, reactive oxygen species, breakdown products of cellular damage. The coral detects these chemical changes and responds. Initially, it tries to help, increasing production of antioxidants, providing more nutrients, trying to stabilize the partnership. But if stress continues, if the chemical signals of algae distress keep intensifying, the coral makes the call, eject them. This is a medical diagnosis and treatment decision happening at the cellular level. The coral is monitoring the physiological state of millions of individual algal cells, integrating that information across its body and executing a coordinated response. Some coral species are better at this than others. According to work by coral physiologist Ross Cunning at the Shed Aquarium, certain corals can selectively expel only the most damaged algae while retaining healthy ones. Others can swap one species of Cymbidium for another more heat-tolerant species after a bleaching event. This is adaptive management of a symbiotic partnership. The coral is adjusting the composition of its internal ecosystem based on environmental conditions, favouring algae that perform best under current circumstances. It's making personnel decisions without a brain. All of this, the chemotaxis of larvae, the predator detection of adults, the competitive assessment between neighbours, the symbiont management within cells, emerges from the same basic machinery. Receptor proteins detecting molecules, triggering signalling cascades, integrating information, producing behaviour. The coral's chemical senses are its primary window into the world, and through that window, it perceives a remarkably detailed picture of its environment, who's nearby, what's coming, whether conditions are improving or deteriorating, whether to fight or retreat or wait. But sensing alone doesn't constitute intelligence. Integration does. The ability to combine different types of information, to weight them against each other, to produce responses that reflect not just single inputs but the whole context. And that's where things get truly interesting. Intelligence emerges where information streams converge. A coral polyp isn't responding to single stimuli in isolation. It's receiving dozens of signals simultaneously. Light levels changing throughout the day, water temperature fluctuating with tides and currents, chemical cues from predators, prey, competitors, and symbionts, mechanical forces from waves and water flow. Each signal carries information, each one matters, but they don't all matter equally, and they don't all matter at the same time. The polyp needs to prioritize. When a predator attacks, feeding behavior should stop. When spawning time approaches, growth should redirect toward reproductive tissue. When bleaching stress begins, energy should shift toward antioxidant production and cellular repair. This is context-dependent decision-making. The same input produces different outputs depending on internal state and external circumstances, and somehow, without a brain coordinating everything, the coral makes these decisions moment by moment, adjusting behaviour to match the changing demands of survival. Let's examine a specific case: feeding behaviour. A coral polyp extends its tentacles into the water column primarily at night. This much we've established. It's a circadian behaviour, timed by the molecular clock. But the actual decision to extend or retract tentacles in any given moment integrates multiple factors simultaneously. Factor 1. Time of day. The circadian clock provides baseline expectation. Night means extend. Day means retract. Factor 2. Water flow. According to research by coral physiologist Kenneth Seedens at the University of Washington, corals adjust tentacle extension based on current velocity. Strong currents bring more plankton but also make it harder to capture prey and increase the risk of tissue damage. Weak currents are safer but less productive. The optimal strategy varies with flow rate, and corals track this continuously, extending tentacles fully in moderate flow, partially in strong flow, and retracting completely when currents exceed tissue tolerance. Factor 3. Food availability. Corals can detect the chemical signatures of zooplankton in the water. When plankton concentrations are high, tentacles extend even during the day, overriding the circadian default. When plankton concentrations are very low, tentacles may remain retracted even at night, conserving energy rather than wasting it on futile feeding attempts. Factor 4. Threat level. If predators are nearby, detectable by their chemical signatures, feeding behaviour is suppressed regardless of other factors. Survival trumps nutrition. Factor 5. Energy reserves. If the coral is starving, if symbiont photosynthesis isn't providing enough energy, feeding behavior intensifies. Tentacles extend earlier, retract later, take more risks. Desperation overrides caution. Each of these factors has its own signal transduction pathway, its own molecular machinery, and somehow they all feed into a final common pathway that determines whether tentacles extend or retract. Research by neuroscientist Cliff Tabin at Harvard Medical School, studying cnidarian behavior, has shown that this integration happens through convergent signaling. Multiple input pathways, the circadian clock, mechanosensors detecting water, flow, chemoreceptors detecting food and predators, internal metabolic sensors, all ultimately affect the concentration of a few key signaling molecules like calcium ions and cyclic AMP. These molecules act as integrators. Their concentration at any moment reflects the sum of all inputs, weighted by pathway strength. When concentration exceeds a threshold, behavior switches, tentacles extend. When it drops below threshold, tentacles retract. It's analog computation performed by molecular concentrations. The coral's body is functioning as a biological calculator, continuously solving a multivariable optimization problem, given current time, flow, food, threat, and energy state. What feeding behavior maximizes survival and growth? And it's not just feeding, every behavior follows similar integration logic. Consider calcification, the process of building the calcium carbonate skeleton. This is energetically expensive, requiring the coral to pump calcium and carbonate ions against concentration gradients to create conditions where mineral crystals can form. The process is tightly regulated, responding to multiple factors. Temperature affects calcification rates. Warmer water up to a point increases rates. Too warm, and rates decline. Light levels matter. Calcification is typically faster at night when it doesn't compete with photosynthesis for resources. Water chemistry matters enormously. Ocean acidification reduces carbonate ion availability, making calcification harder. But the coral also needs to balance skeletal growth against other demands. During spawning season, energy shifts away from calcification toward reproduction. During stress events, calcification may stop entirely, while resources redirect towards cellular repair and survival. According to work by biogeochemist Robert Stenek at the University of Maine, corals adjust calcification rates dynamically, sometimes hour by hour, based on integrated assessment of all these factors. The skeleton grows faster when conditions are optimal, slower when they're marginal, stops when they're dire. The coral is making resource allocation decisions that would challenge a corporate financial officer, and it's making them without conscious deliberation, purely through molecular integration of environmental and physiological signals. But perhaps the most striking example of behavioral complexity in corals involves their interactions with other organisms, not as prey or predators or competitors, but as partners. Coral reefs are ecosystems, and corals sit at the center of vast webs of mutualistic relationships. Beyond their symbiosis with zooxanthellae, corals host communities of bacteria, fungi, archaea, and viruses collectively called the coral microbiome. These microbes aren't just passengers, they're active participants in coral health, providing nutrients, producing antimicrobial compounds, even influencing stress responses. Research by coral microbiologist Rebecca Vega-Thurber at Oregon State University has revealed that corals actively manage their microbiomes. They produce mucus with specific chemical compositions that favor beneficial bacteria while inhibiting harmful ones. They can shift microbiome composition in response to environmental changes, recruiting different bacterial partners for different conditions. When water temperatures rise, for example, some corals increase production of antimicrobial peptides that suppress temperature-sensitive bacteria while promoting heat-tolerant strains. When nutrient levels change, they adjust mucus chemistry to favor bacteria that can process those specific nutrients most efficiently. This is community management at the microscopic scale. The coral is not just hosting microbes passively, it's curating the community, selecting for partners that provide the most benefit under current conditions, and it gets more complex. Corals also interact with larger organisms in ways that require sophisticated behavioral coordination. Many coral species host gobies or damselfish that defend the coral from predators in exchange for shelter. The fish chase away corallivores, animals that eat coral tissue, protecting their home. But the relationship requires the coral to tolerate the fish, to not sting them with cnidocytes when they brush against tentacles, to allow them to shelter within the colony's branches. According to research by marine ecologist Danielle Dixon at the University of Delaware, corals recognize their resident fish through chemical cues. They learn to suppress defensive responses when detecting the specific compounds in their partner fish's mucus. If a stranger fish of the same species approaches, it may get stung. But the resident is tolerated. This is partner recognition. The coral is distinguishing between individuals of the same species based on chemical identity, maintaining long-term associations with specific partners while remaining defensive towards strangers. The memory required for this extends beyond simple habituation. The coral needs to encode the chemical signature of its partner and retain that information for weeks or months, constantly comparing detected signals against the stored template. All of this, the integration of multiple environmental cues, the dynamic resource allocation, the microbiome curation, the partner recognition, constitutes a behavioural repertoire far more sophisticated than simple reflexes would suggest. But we still haven't addressed the most fundamental question. Can corals learn from experience in ways that persist beyond immediate molecular states? Can they modify their behaviour based on past outcomes? The answer appears to be yes. Experiments by coral biologist Tali Mass at the University of Haifa have demonstrated that corals exposed to mild heat stress respond faster and more effectively when exposed to heat stress a second time. This isn't just the epigenetic memory we discussed earlier, it's behavioural. The corals produce more protective proteins sooner, they increase antioxidant levels faster, they regulate their algae more precisely. They've learned. Not consciously, not deliberately, but learned nonetheless in the functional sense that matters for survival. Experience has modified response. Other experiments have shown habituation to mechanical disturbance. Corals initially retract strongly when touched, but if touching continues repeatedly without actual harm, the response weakens. The coral stops wasting energy on defensive reactions to harmless stimuli. This is sensory gating, filtering out irrelevant information. It requires the organism to track stimulus frequency, to associate lack of harm with specific stimulus patterns, to adjust response thresholds accordingly. These are the building blocks of learning: association formation, memory storage, behavioral modification based on past experience. In organisms with complex nervous systems, these processes occur in dedicated brain structures: the hippocampus for memory, the amygdala for fear learning, the cortex for decision-making. In corals, the mechanisms are distributed across cellular networks, encoded in gene expression patterns, epigenetic modifications, and sustained changes in signaling pathway sensitivity. It's learning without a learner, memory without a mind to hold it, intelligence as a property of living systems, not a property of brains. And this brings us to the deepest question. If corals can integrate information, make decisions, remember, learn, and coordinate complex behaviours without anything we'd recognise as consciousness, then what is consciousness for? What does a brain add that nerve nets can't provide? There are problems corals can't solve. For all their sophistication, for all their molecular elegance and behavioural complexity, corals remain fundamentally limited. They can't plan. They can't imagine alternative futures and choose between them. They can't learn from experiences they haven't had. They can't innovate, invent, or reason abstractly. These limitations aren't accidents. They're consequences of how distributed intelligence works. A nerve net processes information in parallel, with signals spreading outward from points of stimulation, integrating as they propagate through the network. This is powerful for certain kinds of problems: rapid threat detection, coordinated whole-body responses, filtering of sensory noise. But it's terrible for others. Consider navigation. A jellyfish with a nerve net can sense up from down using gravity-detecting state assists. It can move toward light or away from it, but it can't remember where it's been. It can't build a mental map of its environment. It can't plan a route from point A to point B that avoids obstacles. Why not? because a nerve net has no mechanism for spatial memory. There's no place where information about locations gets stored and organised. There's no way to maintain a stable representation of space that persists independently of current sensory input. According to research by neuroscientist Malcolm McIver at Northwestern University, this is the fundamental limitation of distributed nervous systems. They're reactive, not proactive. They respond to current conditions but can't simulate alternative scenarios or imagine distant possibilities. In contrast, brains with centralized architectures can do exactly this. The hippocampus in mammals maintains spatial maps, representations of the environment that exist independently of immediate perception. You can close your eyes and still know where you are in a room, can imagine walking from one corner to another, can plan routes before taking a single step. This is cognitive modelling, building internal representations of the world and manipulating them mentally. It requires dedicated neural structures that hold information stable over time, that allow different pieces of information to be combined in new ways, that support counterfactual thinking. Nerve nets can't do this. They have no dedicated memory structures, no regions specialized for abstract representation, no mechanisms for sustained attention or deliberate planning. Corals live in an eternal present. They can respond to patterns that unfold over hours or days through molecular integration, the circadian clock, the lunar reproductive cycle, the seasonal temperature changes. But these are pre-programmed responses, evolved over millions of years, encoded in gene regulatory networks. A coral can't decide to try something new. It can't observe that its neighbour is thriving using a different strategy and adopt that strategy itself. It can't problem solve in the moment when faced with a truly novel challenge. This became devastatingly clear during recent coral bleaching events. As ocean temperatures rose past historical norms, corals responded with their ancient evolved stress responses: expel the algae, wait for conditions to improve, then reacquire symbionts and rebuild. But in many cases, conditions didn't improve quickly enough. The corals starved, entire reefs died. A brain-bearing organism facing a novel survival threat can try new behaviours, explore alternatives, learn from failure. Natural selection operates on behavioural flexibility within a lifetime, not just across generations. But corals can only respond with the behavioural repertoire evolution has already given them. If that repertoire doesn't include a solution to the current problem, they die. This is the fundamental trade-off. Distributed intelligence is robust, efficient, and energetically cheap, but it's inflexible. Centralized intelligence is expensive, vulnerable to damage, and metabolically demanding, but it's adaptable. Research by comparative neurobiologist Nicholas Strausfeld at the University of Arizona has shown that brains evolved independently multiple times. In vertebrates, in arthropods, in cephalopods, possibly in other lineages. Each time, the evolutionary driver was similar. The need for faster processing, more flexible behaviour, the ability to navigate complex environments or predict prey behaviour, or coordinate elaborate social interactions. Brains aren't better than nerve nets in some absolute sense. They're specialised solutions to specific ecological problems. If your environment is relatively stable, if your niche doesn't require complex spatial navigation or social coordination, if you can afford to be slow and reactive, a nerve net works beautifully. It's worked for cnidarians for 600 million years. But if your survival depends on speed, on predicting where prey will move next, on remembering the locations of resources scattered across a landscape, on coordinating with partners who aren't physically connected to you, then you need something more. You need dedicated processing structures. You need a brain. Yet even brains retain echoes of the distributed systems they evolved from. Your gut has its own nervous system, the enteric nervous system, containing roughly 500 million neurons, organized more like a nerve net than a brain. It operates semi-independently, coordinating digestion, regulating blood flow, even influencing mood through chemical signals sent to the brain. Your heart has intrinsic pacemaker cells that generate rhythmic electrical activity without input from the brain. The rhythm is self-sustaining, distributed across the cardiac muscle. Your immune system makes decisions about self versus other, threat versus non-threat, using cell-to-cell communication networks that look remarkably like coral nerve nets. Distributed processing, no central control, emergent coordination. These systems use the ancient logic of distributed intelligence because that logic works. For certain kinds of problems, it's optimal. According to research by systems neuroscientist Peter Sterling at the University of Pennsylvania, even the brain itself uses distributed processing for many functions. Early sensory processing in the retina and spinal cord happens locally, without involving higher centers. Reflexes are coordinated by spinal circuits, not cortical decision-making. Many homeostatic functions, temperature regulation, blood pressure control, respiratory rhythm, are managed by brainstem circuits that operate automatically, unconsciously. The brain didn't replace distributed intelligence, it built on top of it, adding new layers of processing while retaining the efficiency of local, parallel computation for tasks that don't require centralized control. In this sense, corals aren't primitive versions of brain-bearing animals. They're alternative solutions, organisms that committed fully to the distributed strategy and refined it to extraordinary sophistication. Their limitations are real, but so are their advantages. Corals are effectively immortal. Barring predation, disease, or environmental catastrophe, there's no inherent lifespan limit. The oldest known coral colonies are over 4,000 years old. They've been growing, responding, integrating information, making decisions for millennia. Brains can't do this. Neurons are post-mitotic. They don't divide after development. Damage accumulates. Synaptic connections degrade. Eventually, the system fails. Every brain-bearing organism has a finite lifespan. Corals continuously regenerate. Damaged tissue regrows. Lost polyps are replaced. The colony persists as long as some fragment survives to rebuild. This regenerative capacity is a direct consequence of their distributed architecture. There's no critical structure that must be preserved. Corals are also energetically efficient. Brain tissue is metabolically expensive, consuming roughly 20% of human energy expenditure, despite being only 2% of body mass. Neurons require constant ATP to maintain membrane potentials, to pump ions, to synthesize neurotransmitters. Nerve nets are far cheaper. The neurons are simpler, the signaling slower, the metabolic demands lower. For an organism that can't actively hunt, that depends on passive filter feeding and algal photosynthesis, this efficiency is essential. A coral couldn't afford to run a brain. And finally, corals are modular. A brain is a single, integrated structure. Damage to key regions causes catastrophic failure. But a coral colony is a collection of semi-independent polyps, each with its own nerve net, each capable of functioning alone if separated. This modularity makes coral colonies incredibly robust. Storms break them apart, and the fragments continue growing. Predators eat sections and the remaining tissue regroups. No single injury is fatal unless it's large enough to kill the entire colony simultaneously. These advantages, immortality, efficiency, modularity, aren't trivial. They've allowed corals to become ecosystem engineers, foundation species that build structures visible from space, habitats that support 1/4 of all marine species. But they come at a cost: rigidity. Corals can't adapt within their lifetimes to truly novel challenges. They can't invent new behaviours, explore new niches, respond creatively to unprecedented threats. In a stable world, this works. In a rapidly changing world, it becomes fatal. Climate change is happening faster than coral evolution can respond. Ocean acidification is altering water chemistry in ways corals have never encountered. The molecular programs that worked for millions of years are failing. Some coral species are adapting, shuffling symbionts, adjusting calcification strategies, showing surprising resilience. But these adaptations are still fundamentally reactive, still constrained by the evolutionary programming encoded in their genomes. They can't think their way through this. They can only hope that natural selection happens fast enough to favour the lucky variants that happen to carry genes for heat tolerance or pH resilience. Brains wouldn't save them, but brains are what allowed one particular species, us, to recognise the problem, understand its causes, potentially intervene. The irony is profound. The most sophisticated intelligence on Earth is required to save organisms that never needed intelligence at all. That thrived for hundreds of millions of years on distributed cellular processing. that built reefs and ecosystems and planetary carbon cycles with nothing more than nerve nets and molecular communication. But that raises the final question. If corals can accomplish so much without minds, what does that tell us about what minds really are? Perhaps consciousness was never the beginning of intelligence. Perhaps it was a late addition to a process already billions of years old. We've spent this entire exploration documenting what corals can do without brains. They sense their environment with precision. They integrate multiple information streams. They make context-dependent decisions. They remember. They learn. They coordinate complex behaviors across thousands of cells. They manage symbiotic partnerships and compete strategically with neighbors. Every one of these capabilities would traditionally be described as cognitive, as intelligent, as requiring mental processing. Yet there's no mind doing the processing, no consciousness experiencing the sensations, no subjective awareness watching decisions unfold. The coral doesn't know that it knows. It doesn't experience the sensation of tentacles extending or closing. It doesn't feel pain or pleasure or fear. Or does it? This is where the conversation becomes philosophical, because consciousness is notoriously difficult to define and impossible to measure directly in any organism except yourself. You know you're conscious because you experience being conscious. But how do you know if anyone else is? How do you know if anything else is? The classical answer, dating back to philosopher René Descartes, was to draw a bright line. Humans are conscious because we have souls, or reason, or language, or self-awareness. Animals might have some dim form of awareness, but nothing like human consciousness. And simple organisms like corals, obviously not conscious, obviously just biological machines. But this line has been eroding for decades. Research in comparative neuroscience has revealed that many animals we once thought incapable of sophisticated cognition are far more capable than we imagined. Octopuses solve puzzles and use tools despite having brains structured completely differently from ours. Crows demonstrate insight learning and future planning. Even insects, with brains containing less than 1 million neurons, show evidence of attention, expectation, and simple forms of consciousness, according to work by neuroscientist Lars Chitka at Queen Mary University of London. The more we look, the more we find intelligence in unlikely places, and this creates a problem. Where do we draw the line? According to philosopher Peter Godfrey Smith at the University of Sydney, who has spent years studying the evolution of minds, the problem is that we've been asking the wrong question. Consciousness isn't binary, present or absent. It's not something that suddenly appears when nervous systems reach sufficient complexity. Instead, it's a gradient, a spectrum, something that comes in degrees. At one end, you have minimal consciousness, the simple, felt quality of sensation, the basic distinction between experiencing something and experiencing nothing. A worm recoiling from light might have this: raw sensation without reflection, awareness without understanding. At the other end, you have rich, reflective human consciousness, the ability to think about thinking, to model your own mental states, to engage in abstract reasoning and hypothetical planning. Most organisms fall somewhere in between, and the question isn't, is this organism conscious? But what kind of consciousness does it have? This framework transforms how we think about corals. They clearly lack reflective consciousness. They can't think about their own thoughts. They can't imagine alternative futures. They have no sense of self as a persistent entity extending through time. But do they have phenomenal consciousness? The raw feel of experience? When a coral retracts from a predator's touch, is there something it's like to be that coral in that moment? Is there a subjective quality to the sensation, however dim? We can't know for certain. The hard problem of consciousness, understanding how physical processes give rise to subjective experience, remains unsolved. But we can look for markers, for the functional signatures that correlate with consciousness in organisms where we're more confident it exists. One such marker is integration. According to neuroscientist Giulio Tononi's integrated information theory, consciousness arises when a system integrates information, when it combines multiple inputs to produce unified responses that couldn't be generated by the parts independently. Corals do this. Their nerve nets integrate signals from multiple sensory modalities, combining temporal information from circadian clocks with spatial information from chemical gradients, with mechanical information from water flow. The resulting behaviour reflects all inputs simultaneously, not just the sum of independent responses. Another marker is flexibility. Conscious organisms don't just react mechanically to stimuli, they modulate responses based on context, internal state, past experience. Their behaviour shows variability that suggests something more than pure determinism. Corals do this too. The same stimulus produces different responses depending on time of day, recent feeding history, threat level, energy reserves. There's contingency in their behaviour, adaptation to circumstance. A third marker is learning, the ability to modify behaviour based on experience to improve performance with practice. Again, corals qualify. They habituate to harmless stimuli, sensitized to repeated threats, show faster responses after prior exposure to stress. These markers don't prove consciousness. They're compatible with sophisticated unconscious processing. But they suggest that whatever generates subjective experience in more complex organisms exists, at least in primitive form, in corals. Philosopher Thomas Nagel famously asked, "What is it like to be a bat?" pointing out that bats experience the world through echolocation in ways we can't imagine. The question was meant to illustrate the mystery of consciousness, how subjective experience might differ radically between species. We might ask, what is it like to be a coral? And the honest answer is, probably nothing like what it's like to be you. There's no visual experience. Corals have no eyes in most species. There's no auditory experience. They have no ears. There's no proprioception in the way you experience your body's position in space because there's no centralized processing to build a unified body map. But there might be chemical experience, a continuous felt quality of the molecular landscape, something like taste but more fundamental, more all-encompassing. The sensation of food molecules in the water might have a subjective character, a feeling that means nutrition available. The sensation of predator chemicals might feel different, might carry a quality that means danger. There might be temporal experience, a sense of the circadian rhythm, not as conscious anticipation, but as a felt pulling toward different states at different times. The approaching night might feel different from midday in some raw, non-conceptual way. There might be social experience, the chemical presence of neighbouring polyps, the difference in quality between self and other, between kin and stranger. These wouldn't be thoughts. They wouldn't be perceptions in the way you perceive, but they might be something. Dim, distributed, diffuse, awareness without a centre, experience without an experiencer, or there might be nothing. The coral might be what philosopher Daniel Dennett calls a zombie. All the functional behaviour of intelligence with no subjective experience whatsoever. The lights might be on, but nobody home. Just molecular mechanisms executing pre-programmed responses. We can't decide between these alternatives empirically. Consciousness leaves no fossil record. It can't be measured with electrodes. The only way you know you're conscious is through direct experience, and you can't access a coral's direct experience even if it exists. But here's what corals definitely show us. Intelligence doesn't require consciousness. Complex, adaptive, flexible behaviour can emerge from systems with no subjective experience whatsoever, or at most, with only the dimmest glimmer of awareness. This has profound implications. It means that most of what brains do, the information processing, the decision-making, the learning and memory, happens unconsciously. Consciousness, if it exists, is riding on top of vast unconscious processing, like foam on a wave. The real computational work happens below awareness. Your brain makes most decisions before you're conscious of making them. Research by neuroscientist Benjamin Libet showed that neural activity indicating a decision to move occurs several hundred milliseconds before the conscious experience of deciding to move. Your unconscious neural processing is calling the shots. Consciousness arrives late, narrating decisions already made. In this view, what corals reveal is the foundation, the ancient unconscious intelligence that's been solving survival problems for 600 million years. The molecular computation, the cellular communication, the distributed decision-making that works without anyone watching, without anyone experiencing it. Consciousness, when it evolved in more complex organisms, didn't create intelligence. It added a new layer, self-awareness, internal modelling, the ability to reflect on experience and plan based on simulations. But the intelligence was already there, running in the background, doing the real work. Corals are showing us what intelligence looks like at its most fundamental: responsiveness to environment, integration of information, adaptive behaviour emerging from cellular communication. No consciousness required, no awareness necessary. Just life responding to life's ancient imperative: persist, grow, reproduce. And yet, knowing this, watching a coral polyp respond to touch, seeing the tentacles retract in coordinated waves, it's hard not to feel that something is happening in there. That the information flow isn't just physical causation, but something more. That being that coral, if being that coral is anything at all, might have some faint quality, some minimal whatness that distinguishes existing from not existing. Maybe consciousness isn't what we thought. Maybe it's not a special property of complex brains, but something more fundamental. A basic feature of systems that integrate information, that distinguish self from environment, that respond adaptively to the world. If so, then consciousness isn't rare, it's universal, and it comes in countless forms, most of which we'll never understand because we're trapped inside our own particular version, our own thin slice of what it's possible to experience. The coral polyp might be conscious in ways we can't comprehend, or it might be thoroughly unconscious, a biological robot executing genetic programming with no inner life at all. But either way, it's thinking, making decisions, navigating existence with elegance and precision, and that, perhaps, is what truly matters. We began by asking how an animal without a brain could make decisions. Now, standing at the end of this journey, the question itself seems wrong. It assumes that brains are what make decisions possible, that intelligence requires the hardware we carry in our skulls, but corals have been deciding when to feed, when to reproduce, when to fight or flee or wait, since before brains existed. Since before the Cambrian explosion spawned the diversity of neural architectures we see today. Since before eyes opened or blood flowed or the first synapse fired, the coral polyp we started with, translucent and anchored to rock, is still there, still tracking light with its circadian clock, still integrating chemical signals from predators and prey, still coordinating with its algal partners, still building skeleton atom by atom in the darkness. Nothing has changed for the coral, but something has changed for us. We've traced intelligence to its roots, not to neurons but to cells communicating, not to brains but to information flowing through living tissue, not to consciousness but to responsiveness itself, the ancient capacity of life to sense and adjust, to distinguish beneficial from harmful, to persist in a changing world. This is what intelligence looks like without decoration, without the elaborate machinery of perception and memory and imagination that brains provide. Just the essential core: signal, integration, response, and it works. It's worked for longer than forests have existed, longer than vertebrates have walked on land, longer than eyes have seen colour. The coral reefs built by these brainless architects are older than the Amazon rainforest, more biodiverse than most terrestrial ecosystems visible from orbit. We tend to measure intelligence by how much an organism resembles us. Can it use tools? Can it solve puzzles? Can it recognize itself in a mirror? These benchmarks privilege our particular flavor of cognition, our specific evolutionary solution to the problem of survival. But corals are reminding us that intelligence is broader than that. It's any system that processes information about the environment to produce adaptive behavior. It's the bacteria sensing chemical gradients and swimming toward food. It's the plant root growing around obstacles in soil. It's the immune cell distinguishing self from pathogen. It's the coral polyp integrating light and chemistry and time to coordinate the behaviors that keep it alive. These aren't metaphors for intelligence. They are intelligence, functioning at scales and in substrates we're only beginning to understand. What corals teach us is humility. We are not the first intelligent organisms. We're not even the first conscious organisms, if consciousness exists on a gradient as many researchers now suspect. We're late arrivals to a very old conversation, one that's been running in molecular dialect since the first cells learned to sense their neighbours. Our particular gift, the reflective, imaginative, linguistic intelligence that allows us to write and read these words, is remarkable. It's enabled us to reshape the planet, to leave Earth's atmosphere, to decode our own genetic instructions. But it's a specialised adaptation, one solution among many to the challenge of being alive, And in some ways it's made us blind to other forms of intelligence. We've looked at corals and seen simplicity because they don't have brains. We've looked at their behaviour and called it instinct because it doesn't involve learning in the way we learn. We've looked at their responses and called them reflexes because they don't involve conscious choice. But beneath those labels, the coral is doing exactly what we do: sensing, integrating, deciding, acting. The molecular details differ, the speed differs, the flexibility differs, but the fundamental process, information flowing through a system to produce adaptive behaviour, is the same. This matters for more than just our understanding of corals. It matters for how we think about intelligence in general, including artificial intelligence. We're building machines that process information, integrate multiple inputs, learn from experience, make decisions without human oversight. We ask, are they truly intelligent? Can they really understand? Or are they just executing algorithms, following rules without comprehension? The coral offers a third possibility. Maybe understanding isn't binary. Maybe it's not something you either have or don't have. Maybe it's what happens when information flows through any system in ways that produce adaptive responses. Maybe the algorithm is the understanding, the process is the comprehension, the response is the knowledge. If a coral can be intelligent without consciousness, then perhaps intelligence and consciousness are separate phenomena that can exist independently. And if that's true, then machines might be genuinely intelligent long before they're conscious, if they ever become conscious at all. These aren't just philosophical puzzles. They're urgent questions as we navigate a world where artificial systems increasingly make decisions that affect human lives, where we're trying to determine what moral status to assign to different kinds of minds, where we're facing an extinction crisis driven by environmental changes that are challenging organisms' ability to adapt. Coral reefs are dying. Not because corals lack intelligence, but because intelligence without flexibility can't respond to unprecedented change. The molecular programs that worked for millions of years are failing because the environment has shifted faster than evolution can track. We, with our flexible, imaginative, brain-based intelligence, have caused this crisis. And now we face a choice. Will we use that same intelligence to solve it? This is what brains add to the ancient intelligence of cells. The ability to imagine things that don't exist yet, to plan for futures that haven't arrived, to coordinate actions across individuals and generations toward goals that won't be realized in our lifetimes. Corals can't save themselves from climate change. They can only respond with the tools evolution gave them: shift symbionts, adjust calcification, hope that some genetic variants in some populations carry the lucky mutations that confer heat tolerance. But we can imagine solutions. We can develop interventions. We can choose, deliberately and consciously, to preserve these ancient intelligences that have been making decisions in darkness for hundreds of millions of years. In doing so, we'd be protecting more than just corals. We'd be protecting the ecosystems they support, the millions of species that depend on reef structures, the coastal communities that rely on reefs for food and storm protection. We'd be protecting living libraries of biological innovation, organisms that have solved problems we're still trying to understand. But perhaps most importantly, we'd be recognizing that intelligence is not a pyramid with humans at the top. It's a web, vast and ancient, connecting all living things through the fundamental capacity to sense and respond and persist. The coral is not below us on some evolutionary ladder. It's beside us, parallel to us, solving different problems with different strategies, but participating in the same grand conversation between life and world that's been running since the first molecules learned to replicate. We started with a coral polyp, barely visible, anchored to rock in shallow water. We end with the same polyp, unchanged except in how we see it. It's not simple, it's not primitive, it's not a failed attempt at becoming something more complex. It's a success, a solution, an intelligence that's been working since before brains existed and will likely persist long after the last human brain stops firing. The polyp doesn't know we've been watching. It doesn't know it's taught us anything. It's simply doing what it's always done, sensing, integrating, deciding, persisting, living without a brain, thinking without a mind, remembering without memory, proving that intelligence was never about the hardware. It was always about the conversation between inside and outside, self and world, signal and response, and that conversation is as old as life itself.